Okay, so thank you very much for being still here despite the late time and the hot in this room. So it will be actually very short, and I don't have any fancy video to show you as my previous speakers. So as you can guess from the name, the BraiNAVigation Lab has to do with brain. So how the brain function is structured and work. So in a sense, this gives you a flavor of the way the AI is pervasive right now. Life sciences have a strong need of artificial intelligence, and is one of the fields where explainability and interpretability is a must. You cannot conceive a very fancy and very well-performing algorithm which you are not able to explain how it works to the doctors, because this will avoid its exploitability actually in the clinics. This is our lab. We are quite a bunch of persons that you probably have already seen here around in the different courses. And now let me go a bit into the details. So we have many collaborations all around the world, both in Italy and also in Europe, but in the US and in Brazil as well. As far as I am concerned, I did my degree in engineering, electronic engineering in Milano, at Politecnico di Milano. And then I had a PhD at the Swiss Federal Institute of Technology in Lausanne. So I don't go into the details of all these collaborations, but they are all centered about neuroimaging. Let's now see what the neuroimaging means by starting having a look to our labs. We have a dedicated lab, which is the BraiNAVigation Lab, where we have some equipments that allows us to perform some of our research. For instance, we have EEG recorders, so the recorders that allows to capture the signals that are on the scalp of a person while doing a given task, or also while resting. We also have some other facilities that are used for BCI, so brain-computer interfaces, which can be used both in clinics and in industrial environments, for instance, for monitoring the fatigue into the subjects that are doing a given task in the industry, or to help in rehabilitation of people. Then we also work with MRI, magnetic resonance imaging, which is the main business of our activity, together with EEG, and that we acquire either in collaboration with the hospital here in Borgo Trento, in Borgo Roma, or also through our collaborations around the world. So the main difference with respect to what you have seen before is that we look into the subject instead of looking around into the environment. But the main issues are the same, the main technologies are the same. And on top of this, we must be able to explain what we observe, what we find, and what we predict. So here is a simple representation of all the data that we are dealing with. So we have, as I was mentioning, imaging data that are called neuroimaging because they concern the brain, actually, that could represent both the brain structure and function. What does it mean? The brain structure means that you investigate the texture of the brain. And the function means that you look how the brain performs, is activated or deactivated in specific conditions. You can also use another type of magnetic resonance imaging to understand how the different regions of our brain are wired. So how one region is connected to another. This is called diffusion MRI, and basically allows you to reproduce the path of the white matter axons in our brain. So this is for imaging. Then we also deal, as I was mentioning before, with EEG data, which represent another way of studying the brain function, but also with genetics. The trend now is to try to link the features of our brain at different levels, from the microscopic level, so the genes, to the image level, okay? So as you can guess here, there is a wide diversity in the data. We have data that are multi-dimensional, from 1D to 3D, 4D, 4D plus time, and so on. We have the problem of missing data, because maybe for a given subject, you have one modality, but you don't have one other. We have a very different scale among the data, so we have to be able to investigate the relationships within all these different aspects of our being, actually, of our brain. Then this, of course, also calls for other techniques. For instance, graphs are used to represent what we call connectivity, structural or functional connectivity. Now I don't have time to go into the details of what it is, but actually this explains how the different regions are not only connected, but are synchronized or desynchronized. This is applications both in neuroscience and in clinics. So there is really a strong need to deal with this data. Machine learning is very important. Deep learning is even more important, because now we have a very wide variety of data that are big data that require this kind of structure to be effectively processed. Of course, okay, this is not visible there. We can do different things with this data, studying associations between the different kind of data, doing classification, prediction, multimodal fingerprinting, and so on. So in all this, the emphasis will be on methods that are explainable, so can be explained and can be interpreted, so that we can communicate to the doctors or to the neuroscientists what we have obtained with our models. So the why and the how of the outcomes of our algorithms. This, of course, spreads across very different methods, and we don't have time now to go deeply into these aspects. But just to give you an idea of how it works, I mentioned here one application where it is particularly simple to understand what is the point for us. For instance, here we have a CNN that tries to distinguish two different phenotypes of a disease that is, in this case, multiple sclerosis. And the two phenotypes distinguish between those patients that are going to get worse, so to develop the disease, and those that will be stable. Of course, the sooner this difference is detected, the better it is, because the treatment is different, because the prognosis is different. So what we did was to extract some microstructural descriptors from our data, in this case, diffusion MRI. So we have these maps. With these maps that are 3D data, we fed this 3D network, and we got a classification, okay? Then we used a model that it is the layer-wise backward propagation model, which allows you to build a heat map in the input data that tells you where the decision of the classifier were based. Okay, so you can identify the region of the brain where there is the main difference between the two categories of patients. So now, of course, you get the result, but you have to understand whether it is correct or not. So one way is to discuss with the doctors, and this is, of course, what we did. And other ways is to take these images and see whether the regions where we get the main contributions actually make sense from a point of view of the disease. And this was actually the case. So this is just an example to explain you why it is important for us to explain the result, and one simple, very simple way to use it. We are extensively working on the explainable AI methods applied to the biomedical field, and in particular to the neuroimaging field. We had some very recent publications in this respect. And we also have a special session on explainable AI in biomedical image processing at the incoming ICIP conference to be held in Bordeaux after the summer. ICIP is the International Conference on Image Processing. So I don't go deeper into the topics of our research. If you are interested, you are more than welcome to come and to visit our labs. As Professor Giacchetti mentioned, we are strongly connected with the VIPS lab. And this tree that has the root on me actually is connected to the VIPS lab as well. The contents that you will find in this AI master program that are related to our activities are, of course, those related to explainable AI, where I will be one of the teachers, in the visual intelligence one, and are, of course, connected to machine and deep learning, computer vision and deep learning, and intelligent interfaces, as it is easy to guess. So thank you very much, and have fun with this new master degree.